Okay, for our whirlwind review tour, we're gonna do a very quick um, replay of microscopes and the very introduction of cells. Basically, a microscope is an imaging system. It takes something very large and makes larger images out of it. This one says, do your stuff, you're on Microscope. The important aspect of any imaging system is clarity. Clarity is expressed as resolution. Resolution classically was measured by figuring out what this tiniest gap was that you could see was still a gap. Now my fingers are not quite touching, but I think on the screen it looks like they are touching. We've hit the resolution power of the screen that I'm looking at. And screens have resolution, very often measured in pixels. But again, the denser the pixels, the more you can see a gap as a gap. In microscopes, you've got your two major imaging systems. One uses a light beam, nice and cheap, glass lenses, easy to use in a microscope, easy to train people on. But when you get up to about 1200 magnification, light beams have a wavelength, right? We talked about that for photosynthesis, which means they have a wave height. And the smallest gap that you can start to resolve is actually getting the size of the beams of light that are making the image. So something gets closer together, it just blocks the beam of light, and you can no longer see a gap. It looks like those two objects are continuous. So there are some computer manipulations that allow resolution to go beyond 1200 for light microscopes. There's a link in the book to one of those developed, I think, at Harvard. Originally developed with processes for space probes. Space probe cameras packed, in a very weight restricted probe. They found ways to fiddle with subsequent or successive images and use those successive images to increase the resolution of the overall picture. And the Harvard thing is basically applying that same sort of logic. But typically, even for our microscopes top out at about 1,000, and even then, the resolution isn't great. The older microscopes actually have better lenses. You've got two ways to process the imaging beam. You can pass the imaging beam through your object, transmit it, that's a transmission microscope. You can bounce the beam off of the object, that's a scanning scope. It used to be called a reflective microscope, but for electron beams, you actually scan the beam across the surface and pick up the reflections. Now, because the beams are scattering off of a surface, the resolution on a scanning scope is typically worse than the resolution on a transmission scope. You got the beam coming through, and the beam does scatter a bit going through what you're looking at, but there's more beam to collect, and so you tend to get better resolution. Now, having said that, there's some very bizarre scanning scopes. They use a teeny tiny probe that actually physically moves across the surface. And the link in the book that actually shows you atoms as bumps on a surface is what they call a scanning tunneling microscope. Very, very expensive. Great to be able to see basically the outer electron cloud of an atom, but doesn't look like much. Looks like a bump. These are pictures from a scanning electron micrograph. Processing. For a transmission scope, you need the beam to come through what you're looking at, which means what you're looking at has to have some level of transparency. Can't be completely transparent or you wouldn't be able to see it. So you have to adjust the transparency of your object so that you get some contrast in what you're looking at. In the case of a light microscope, you're physically registering that stuff in your eyes. For an electron microscope, you're hitting the beam is hitting a screen, or it's being collected in a camera, and then projected on a screen. But you got to be able to get some of the beam through, but not all of the beam through. For small objects, this is more true for light microscopes, you may need to do what's called clearing. You want to look at a flea under a transmission microscope. If you put a flea on a slide and put it under the microscope and then look at it, you see a wonderful silhouette of a flea, because the flea is opaque. The light's not coming through it, unless you put it into a chemical that sucks a lot of the dark pigment out of the flea. It makes it semi-transparent. And then on a slide, you can see structures within the silhouette. You can see where the legs are, you can see the bristles, you can see other sorts of things. So intact whole objects very often are cleared. For things that are not whole objects, that you're looking at pieces of things, you typically are going to look at thin slices, what are called sections. And there you need to do sectioning. You take a biopsy. Somebody takes a cylinder of tissue out of a breast lump or who knows what. It goes to the lab, it goes into preservative, because it's going to immediately start rotting if you don't preserve it. Then it is suspended in a material that will soak into it and hold it together when you slice it really thin, because preserved tissue will crumble if you try to slice it really thin unless it's held together. For light microscopes, that's typically done with wax, and then you can take the slices and dissolve the wax away, just leaving the slice of tissue, which then needs to be stained, because the thin slice of tissue doesn't have great contrast in it, and it's hard to see things. So what we do is we throw a bunch of varied dyes onto the slice, and some chemicals pick up a green dye, some chemicals pick up a purple dye, some chemicals pick up a red dye, and it makes a section very, very readable for a light microscope. For electron microscopes, you can't slice wax thin enough. So it is suspended in a hard plastic. Very, very small objects in a very, very small block of hard plastic that is then sliced with a knife that's literally a piece of broken glass. You get an extremely thin piece of section. You have to leave the plastic on there because there's no way to get rid of it. But the plastic doesn't interfere with an electron beam. You take the slice and you put it on a grid, going across the space in the grid, and then you have to stain with an object that interferes with an electron beam. Typically osmium tetroxide, a heavy metal that soaks into biological materials at different amounts. Is that some things absorb it completely and then the beam doesn't come through and you see black. Sometimes the beam comes through partly and you see gray. Places where there's spaces, you see white on the screen. Electron microscopes are more difficult to work with. They're more expensive. There's way more controls. An electron beam is a very antsy sort of thing that scatters very easily, so you need a complete vacuum inside what they call the column that the beam travels through. And osmium tetroxide is a highly toxic material that can poison you if you're not careful with it. Any object that's going to go in whole, which happens in scanning electron microscopes, you may put a whole object in and bounce the beam off of it, then it needs to be completely dried out. Otherwise, water will evaporate from biological tissue into the vacuum and mess everything up. And then, because biological material will absorb an electron beam to some extent, you need to coat the object with gold or platinum, just literally an atom's thickness of it, to reflect the beam. But the whole processing is much more expensive for electron microscopes. And so schools don't buy a whole bunch of them. Is that our electron microscope lab had one good transmission scope and one good scanning scope. Our school here has a scanning scope that isn't really appropriate for biological materials. They use it downstairs in the clean room to look at chips. So you don't find a lot of those out there. When it comes to sectioning, if you have an object that has orientation, Like say you're gonna look at an arm. You can slice it this way. That's a cross section, sometimes called a transverse section, often labeled as an X section. If you're going to go, well, take a whole body. If you're going to slice this way, lengthwise, going in from right to left, that is a longitudinal or a sagittal section, S-A-G-I-T-T-A-L, sagittal. If you're going to slice this way, kind of, feed your thing in, and so the first thing that would come up would be my nose and unfortunately my belly, a little bit too much of it there. That would be a frontal section. So that one's an easy one to remember. And anything that comes in at a weird angle is considered to be an oblique section. Orientation can be very important to know when you're trying to figure out what you're looking at. When I used to teach embryology in the lab, what the students looked at were what are called serial sections. An entire embryo was fed into the knife in a certain orientation. So there were slides that were cross sections from the top of the head to the bottom of whatever the bottom was of an embryo. Some of them were transverse sections, sliced this way. We occasionally have frontal sections. And they needed to be able to look and orient themselves and know whether they were looking at liver or heart or kidney, and knowing what angle the slice is at is often a very, very important thing. When you're working with light microscopes, there are ways to play with the light in order to help yourself see things. When you're trying to look at live objects that are actually active, like protozoans and such, then they're fairly transparent and not easy to see. So there are microscopes, I've got one on the table over here, it's the only one that we've got, that's called a phase contrast microscope. The beam is actually hollow. And so what you're really hitting the objects with is a ring of light, and the way that it tends to scatter off of transparent objects, it tends to give the edges kind of an aura. You can see things better. There are some light microscopes where the light comes in from the side and reflects off. It's not really a scanning scope, but it's not completely a transmission scope. But you're seeing lit up objects against the dark background. That's what they call a dark field. There's interference microscopes where you're actually hitting certain chemicals with a particular frequency of light to make them fluoresce. There's a whole bunches of tricky things that people have done with microscopes to make stuff show up that's not easy to see with a regular microscope. As far as cells, we're going to be getting into cells, right? Cells are a really big deal. Just going to go quickly through the classic cell theory of Schleid and Schwann from the 1830s, I think. Yeah, from the late 1830s. So this is a really old theory that's held up very, very well. But it does get in the way of some things. To them, all living things were made up of at least one cell. You could have just one cell, a lot of unicellular organisms. You could have multi-celled organisms. They didn't really understand about colonial organisms, but unicellular colonial organisms the way that we do now. But they thought that all living things had to be cellular. This gets in the way of defining viruses as living things. That they decided that the cells only come from other cells. They didn't at the time realize that cells had to come from cells that were related to them. For a while they thought that sick people generated bacterial cells because they found bacterial cells in infections and in certain diseases and stuff. But they didn't realize that there was a form in which they could go from person to person. I forget what Pasteur's micro theory wound up being. Drawing a blank on that. Sucks to get old. They decided from a labeling standpoint that the smallest thing that they were going to consider alive was a cell, even though they could already see things inside a cell that had its own activity, that there were chambers in there doing things. They said, okay, those are not alive, not unless they're cellular. Later on, we found that there are cells that live in other cells, like malaria, but I don't think they knew that at the time. And then basically that cells show more similarities than differences, which, as I've told people before, is more of a kind of a mission statement for research scientists as a way to say what I'm doing is valuable. Is yeah, I'm looking at these weird little cells that nobody cares about, but the stuff I find out is applicable elsewhere and maybe can help cure cancer. They always want to cure cancer, which itself is a silly thing to say, as you'll find out when we get to the cancer chapter. In terms of cells, cells are surrounded by cell membranes. That is a characteristic of all cells. There's a membrane, a lipid-based membrane, a phospholipid bilayer. We'll talk about this later. There may or may not be cell walls. There may be a construct around the membrane of various materials that itself is not considered to be a part of the living cell, but is certainly a very important part of keeping the cell living. Cells are very small. They fall within a fairly narrow range of sizes. You don't tend to find cells below that size range or above that size range. Below is pretty easy to explain. A cell has a certain minimum number of things they need to have happen inside that space in order to be alive. And the cells that are below the threshold, the small threshold, are cells that are intracellular parasites. They live in other cells, and so they don't need to do everything because the surrounding host cell does a bunch of stuff for them. Larger gets a little bit trickier. The old math thing that as the cells get bigger, the volume increases faster than the surface is a classic explanation, but unfortunately that only, it's like the old physics expression, assume a spherical cow. No, is assume a spherical cell. That's what they've done. And for spheres, the math relationship holds up. Most cells are not spherical. And probably the explanation for why large cells don't exist is that it's much more efficient to be multi-celled than to be a giant single cell. So just from a competitive standpoint, is that if there were giant single cells at some point during the development of life, they got outcompeted by multi-celled systems. First colonial and then pure multi-celled systems. One thing that's useful, and you'll see this as we go through, is that a lot of what we've discovered inside cells and in the technology of seeing things in cells has been developed since the 1950s, which means a tremendous amount of this was done in the United States and England. And so older terms tend to be very Latin-y. Mid-range terms tend to be a weird mixture of Latin and English, like microfilaments. And then a lot of later stuff is more, just English-based, and a lot of times pretty easy English to remember. So that's helpful when you're memorizing terminology for cells. Okay, as I said, quick review. That's it for this chapter.